Good day and welcome to our course on basic food microbiology. Let's have a look at our course outline. Firstly, we will be identifying different types of microorganisms. Secondly, we will understand the main structural components of different types of microorganisms. We will also demonstrate an understanding of the microbial growth curve. We will identify the growth requirements of microorganisms. We will understand the main control measures for microbial hazards in food safety, and then we will learn a little bit about the main foodborne pathogens. After completing this course, you will have a good understanding of biological food safety hazards. You will be able to implement good manufacturing practices to prevent microbial growth. You will be able to identify control measures to limit biological contamination as part of a hazard plan, and you will be able to contribute to solving problems and implementing corrective actions relevant to microbial spoilage and contamination. Firstly, let's understand what is a microorganism. A microorganism is a living organism or a living being that's too small to be seen with the naked eye. We use an instrument called a microscope to enlarge microorganisms so that we can see them. Oftentimes we forget about microorganisms because we cannot see them, but just because we can't see them doesn't mean that they are not there. There are also different types of microorganisms. The three types that we will be looking at during this course is bacteria, fungi, which includes yeast and molds, and also viruses. There are more microorganisms, but in terms of food safety, we are going to concentrate on these. Microorganisms can be found all around us and live in the water, soil, and air. They are literally all around us. Microorganisms also form part of the human body and play a very important role in gut health. So it's very important to remember that not all microorganisms are bad. There are a lot of microorganisms that are very beneficial, but then there are also those that can cause food spoilage and food poisoning. Let's have a look at some structures of the bacterial cell. Now on your right, you will see a typical bacterial cell. Bacteria are unicellular organisms. In other words, they consist out of one cell. A typical bacterial cell will contain DNA in its nucleoid. The function of the DNA is to control life and reproduction. Without the DNA, the cell is not alive. The bacterial cell also has a thick cell wall. The cell wall is there to give the bacterial cell its shape and to provide protection. The cell membrane is a thinner layer that is found on the inside of the cell wall, and this allows dissolved substances to enter and exit the cell. In the picture on your right, you will also see little hair-like structures on the surface of the bacterial cell. These are called pili, and they are there to help the cell attach to a surface. Some bacterial cells also have a long tail-like structure called the flagellum, which is there for movement. Bacterial cells also come in different shapes and sizes. As can be seen from this picture, we get bacterial cells that are rod-shaped, and they are called the bacillus. There's also the spherical-shaped bacterial cells, which are called the coccus. The spiral-shaped bacterial cells are called spirilla, and the curved-shaped are called the vibrio. These shapes are often used to identify different types of bacteria, and that is also used in the bacteria's name, like Bacillus cereus or Staphylococcus aureus. Bacteria are also classified according to their oxygen requirements. Some bacteria grows in the presence of oxygen, and they are called obligate aerobes. And that's because they need oxygen to grow. Then we get the obligate anaerobes. They grow in the absence of oxygen, as can be seen in this picture of the test tube. They will grow at the bottom of the test tube, away from the oxygen. We also get facultative anaerobes. These bacteria can grow in the presence or the absence of oxygen, but they grow best in the presence of oxygen, as can be seen in this test tube sketch. The aerotolerant anaerobes grow in the presence or absence of oxygen, so they are actually not affected by the oxygen concentration. The microaerophiles grow where a small amount of oxygen is present. So from this slide, you can see that different types of bacteria have different oxygen requirements for growth. Bacteria can also be classified according to their temperature growth requirements. There are bacteria that grow optimally at colder temperatures. These are called your psychrophiles. A large group of bacteria like to grow at body temperature. These are called the mesophiles. Then there are bacteria that can withstand high temperatures, and they are called the thermophiles. Some bacteria can grow at very high temperatures, almost boiling point. These are called the hypothermophiles. So different types of bacteria have different optimal growth temperatures. This becomes very important when we start looking at food spoilage, because some bacteria will grow optimally in refrigeration conditions, and some bacteria can even resist cooking temperatures, like your thermophiles and your hypothermophiles. More about that a little bit later. It's also important to know how bacteria reproduce. The process by which bacteria reproduce is called binary fission, which basically means splitting into two. As you can see in this diagram, one cell will start to grow and will start to elongate, especially in its genetic material. Then there will be a division in the genetic material, and a septum or separation will start to form. This is when the cell will start to split in two and then eventually form two daughter cells from the one mother cell. If growth conditions are just right, in other words, there's enough food for them, there's moisture, the temperature is just right for bacterial growth, this process of binary fission will take place every 20 minutes. So one bacteria cell can eventually split to form thousands and millions of bacteria cells if the condition for growth is optimal. Let's have a look at yeast cells. Yeast cells are very similar to bacteria cells in that they are unicellular. They are a little bit bigger than bacteria cells. They also have a nucleus with the DNA, which also controls the life and reproduction. They also have a cell wall, which is there for shape and protection of the cell. It also has a cell membrane that will allow dissolved substances to enter and exit the cell. Yeast cells reproduce by a process called budding. As you can see in the picture on your right, these small buds form on the surface of the yeast cell and will eventually break off from the mother cell, and that will then be another individual yeast cell. This is called budding. Molds, on the other hand, are multicellular organisms. In other words, they consist out of more than one cell. Molds also have what is called hyphae, which absorb nutrients and transports it throughout the mold. It also anchors the mold within the substrate that it is growing on. Molds also have a sporangium, which is a sac-like structure that contains the spores, and the spores are there for reproduction. If you look at the picture of mold growing on bread, you will see that the hyphae anchors itself in the bread to take up the nutrients, and that the sporangium will release the spores. Air movement will cause these spores to be distributed through the air, and wherever those spores then land, if the conditions are favorable, it will germinate to form a new mold and therefore spread throughout an area or within a facility. Of particular importance to the food industry is a virus called the bacteriophage. Now, the bacteriophage is a virus that attacks bacteria cells. Now, a virus always requires a host cell to replicate and survive. So without a host cell or without a bacteria cell that it can grow in, it cannot live. Now, a virus has a capsid head, which is the protection for its DNA or RNA that is inside, which is the genetic material. It also has spikes at the bottom where it attaches to the cell, and tail fibers and a base plate that will inject into the cell and pump its DNA into that cell. So in terms of reproduction, you can see that a virus cell will attach itself to the surface of a bacteria cell and then inject its DNA into the bacteria cell. The bacteria DNA gets destroyed and the virus DNA takes over the control of the cell. It then forces the bacteria cell to produce virus genetic material, and eventually the cell will burst and release more viruses to the environment. And in that way, the virus basically hijacked the bacteria cell and used it for its own advantage and its own reproduction. So what is the economic importance of food microbiology? Microorganisms can be used to our advantage. For example, during food processing, we use microorganisms to produce food such as wine, beer, bread, cheese, etc. It can also be used for food preservation and for the production of probiotics, which is very important for good gut health. There are various microorganisms that live inside the human gastrointestinal tract and needs to be there in order to ferment your food. A disadvantage of microorganisms are that they are responsible for food spoilage and a lot of food waste. Some microorganisms can also cause foodborne diseases, actually result in illness and even death. Let's look at the growth curve of microorganisms. This is basically the pattern that microorganisms grow in over time. So you will see here that microorganisms follow a growth curve with 4 phases. The first phase is called the lag phase. The second phase is the log phase. Then we get the stationary phase, and then we get the death phase. Let's look at each one of these phases of the growth curve. Now, the first phase is called the lag phase. Now, during the lag phase, cells start to adapt to a new environment. There's no increase in the number of cells, only an increase in the size of the cell. So the cells basically establish themselves. Proteins required for growth is synthesized, and any cells that has been injured or damaged during the shock of being placed in a new environment is repaired. So during the lag phase, you will see there's not really an increase in the number of cells, but it's almost as if it's a preparation phase. After the lag phase, you will see that there's a sharp increase in the number of bacteria cells. We call this the log phase or the exponential phase. This is where cells start to divide by binary fission. The number of cells increase exponentially, and there's a constant increase in growth. This is called the log phase. During the stationary phase, the growth rate starts to slow down until it becomes almost static. This is because the nutrients start to get depleted and the amount of waste material produced by all of these bacteria cells are starting to increase. So now there's not enough food anymore and the amount of toxic waste materials are becoming more. The last phase is called the death phase. Cell death starts to occur as there's no more food left for the microorganisms and the amount of waste products just accumulates and the number of bacteria will start to decline. This is called the death phase. So microorganisms move through this typical growth curve with the 4 different stages. Let's have a look at the growth requirements for microorganisms. There are other growth requirements as well, but the most important ones are listed here. Firstly, just like all living organisms, they require food to grow. The second requirement is water. Microorganisms require moisture to grow. Different microorganisms have different moisture requirements. Some need more water, some can grow in less water, but water is of vital importance for all microorganisms. Correct temperature. Remember, we talked about the different optimal temperature ranges for different types of microorganisms. So there are optimal temperature ranges for these various microorganisms. Also, the correct pH. Some microorganisms can grow at lower pHs, where others are very sensitive to pH and cannot grow in a very acidic or a very alkaline environment. Then there's also oxygen requirements. Remember that some microorganisms grow when oxygen is present, and some will only grow when oxygen is absent. So there are various oxygen requirements as well. There are more growth requirements, but these are the major ones that will determine whether a microorganism will firstly survive and grow and how fast they will grow in a certain environment. Let's have a look at some of our main pathogens. Now, pathogens are disease-causing microorganisms. So these are the ones that can cause food poisoning, can cause illness, and can also lead to death. The first one we'll be looking at is Listeria monocytogenes. Now, Listeria is a microorganism that needs quite a lot of water activity to be able to grow. So it has a 0.92 water activity. So it will grow in foodstuffs that are quite moist. Their optimal temperature range is between 4 and 45 degrees Celsius, so they would be classified as mesophiles. For their pH range, they can grow between 4.2 and 9.5 pH. So that is quite acidic and quite alkaline. And also, they can grow in quite a high salt concentration. So even if a product contains quite a lot of salt, this microorganism will still be able to grow. So the fact that it can grow at quite a low pH and because it can grow in conditions where there's quite a lot of salt, these two factors make the organism particularly resilient. Now, Listeria monocytogenes are of particular concern in our ready-to-eat products. Ready-to-eat products are products that will not be heated before eaten, so like a ready-to-eat salad or cold meats. Listeria can also survive freezing temperatures and they can grow at low temperatures. So even if you keep the product chilled, that will not guarantee that Listeria will not grow and multiply. Now, Listeria is also known to form biofilms. Now, a biofilm is a protective layer that the bacteria produces to protect itself against environmental factors. Oftentimes, this makes the bacteria more resilient and it's harder to clean and to remove the bacteria from a surface. Fortunately, Listeria is very quickly killed through heat treatment. And this is required to reduce pathogens to these acceptable levels. So for instance, for milk, we will pasteurize the milk, which is a heat treatment to kill any listeria that might be present in the product. The foodborne disease caused by Listeria monocytogenes is called listeriosis. Symptoms of listeriosis are fever, muscle aches, nausea, and diarrhea. Food products that have been implicated in listeriosis outbreaks include French bologna and salad leaves. So both of these products meet the growth requirements for Listeria. Therefore, Listeria will be able to grow and multiply in these products, and there's no heat treatment that takes place that will kill this pathogen before we eat it. So how can we prevent something like Listeria in our food products? Our first control measure for controlling Listeria would be raw material quality control. Remember that oftentimes Listeria comes with the raw material itself. In other words, the raw meat you buy or the water that you use in your factory. You can ensure that the raw material is free from this pathogen or within certain limits by looking at the certificate of analysis. And this needs to be within suitable levels. You can also do spot checks and test some of your raw materials to make sure that it does meet the specification. Processing is a very good way of killing Listeria. So check your CCPs and your OPRPs, especially if they are killing steps for Listeria. Ensure that meat is thawed correctly, that it is temperature controlled and does not stand for prolonged periods of time at temperatures where Listeria can grow and multiply. Ensure that your product is cooked at the required time and temperature. The cooking instructions must be validated so that it ensures the killing of any possible Listeria that might be present. Ensure rapid cooling after heating so that the product does not remain in the danger zone for too long. Hygienic conditions is very important, so handle the product aseptically and ensure that you maintain the cold chain throughout the entire process. Cleaning and sanitation is, of course, a very important step, and it needs to be done properly and implemented correctly. Ensure that you use good quality cleaning and sanitizing agents, and make sure that those sanitizing and cleaning agents will actually target those types of bacteria that are commonly present in the food product. Another pathogen that we need to look at is called Salmonella. You'll see that it also has a quite high water activity. It has a temperature range between 5.3 and 45, but grows optimally at body temperature, 37 degrees Celsius. It also has a pH range of 4.1 to 9, but it grows optimally between 6.6 and 8.2. So that's a fairly neutral pH range. It can grow in salt concentrations of less than 9%. Salmonella is also sensitive to heat. So anything above 60 degrees Celsius that is maintained for 15 to 20 minutes will destroy any Salmonella present. So heat treatment is a very good way of making sure your product is free from Salmonella. So apply the correct time and temperature combination to ensure that this pathogen is eliminated. Ensure that you store your product at temperatures below 4 degrees Celsius, where any potential Salmonella will not be able to grow and multiply. Ensure that you've got good personal hygiene practices and good manufacturing practices in your facility. And ensure that your water is effectively treated, that there's no contamination with sewage water or any other wastewater. Prevent cross-contamination, especially between raw and cooked food. Cross-contamination is when the bacteria from a raw food product is transferred to a cooked food product. And this can happen through the use of the same knife for raw and cooked meat, or by using the same chopping board that you use for raw meat, you now use for chopping salad ingredients. That will be an example of cross-contamination and can be very dangerous. It's important to ensure that you keep raw materials and cooked food separately, and that is to not cross-contaminate between these products. The foodborne disease from Salmonella is called salmonellosis, and it can cause gastroenteritis, enteric fever, and invasive systemic diseases. Staphylococcus aureus is another well-known human pathogen. It has a lower water activity between 0.83 and 0.92. It grows at a temperature of 4 to 46 degrees Celsius, but it's also a mesophile, so the optimal temperature range would be 37 degrees Celsius. Staphylococcus aureus can grow in a pH range between 4 and 9.8, but optimally it will grow at 6.5 to 7.5, which is fairly neutral pH. The salt concentration is less than 15%, which is quite high, so it can survive in products that have quite a lot of salt. Now, Staph aureus is an organism that is also found in the human body, especially the nose, the mouth, the throat, the eyes. So if you do not have good personal hygiene, Staph aureus can very easily cross-contaminate into a food product. We will control this by implementing sanitary practices in the workplace, which include hand washing, wearing of PPE, and good personal hygiene practices like not sneezing over food and not working with food when you are sick. Ensure good personal hygiene practices to prevent cross-contamination and store food products at low temperatures where the Staph aureus will not be able to grow and multiply. Staph aureus is a human pathogen. It can cause things like sepsis and skin abscesses where there is a staph infection. Another pathogen is called Escherichia coli, or E. coli in short. It also has a quite high water activity and will also grow at a temperature between 4 and 45 degrees Celsius, but will grow optimally at body temperature. It grows optimally at neutral pHs and cannot grow in high salt concentrations. E. coli is commonly found in the gut of humans and warm-blooded animals. The presence of E. coli can indicate fecal contamination. So, for instance, where you work with animals on a farm and you can get cross-contamination with their feces, you will find a lot of E. coli present in the meat of that animal. E. coli is luckily destroyed through heat treatment, but if heat treatment was not successful, you will still have E. coli present in that product. Unsanitary practices on farm level or in processing facilities can cause E. coli to grow and multiply. The problem with E. coli is that it produces a toxin called Shiga toxin. Now, the bacteria can be killed through heat processing, but if it has produced a toxin, that toxin is heat stable and it can still cause infection even after the bacteria itself has been killed. And symptoms will include watery diarrhea, sometimes including bloody diarrhea, a low fever, loss of appetite, nausea, vomiting, stomach pains and cramps, and fatigue. We can prevent our products from containing E. coli by processing it at temperatures higher than 70 degrees Celsius. We can also implement good sanitary protocols in the workplace and avoid cross-contamination between raw and ready-to-eat food. We must apply good personal hygiene practices and avoid unpasteurized milk and juices. Coliforms are a group of microorganisms called the indicator organisms. They are like family of the E. coli organism. High numbers of coliforms in your product can also indicate the presence of some other pathogenic bacteria. So they are almost like the warning lights or the warning signs. If they are in high numbers, there's good chance of pathogens being present in your product. So now that we know all of this information about microorganisms, what can we do to control these biological food safety hazards? And that brings us back to the growth requirements. We have to create an environment that is not favorable for the microbial growth. Food, water, temperature, pH, and oxygen requirements. If we know what the microorganism needs to grow and multiply, then we can control their environment so that they cannot grow and multiply by taking away these growth requirements. The first and one of the most important ways in which to control microbial growth is to remove the source of food. And we do this by cleaning. Cleaning removes the source of food for the microorganism. Sanitizing kills the remaining microorganisms and makes the surface unfavorable for microbial growth. And that is why it's important to clean and sanitize all food handling equipment. We must also maintain good personal hygiene and establish rules regarding sneezing, coughing, licking of fingers, chewing, and hand washing. By doing these things, we remove the source of contamination and the nutrients that the microorganisms need in order to grow. If there's no food and the surface is not favorable for them to grow on, they will not grow there. Another growth requirement is water. So we can remove moisture from a product, and we do that through dehydration. Now, dehydrating food helps to preserve the product since the water activity is too low for bacteria to grow. Now, remember that our pathogens all require quite a high water activity. In other words, they need moisture to grow. So by taking the moisture out of the product, we prevent the pathogen from being able to grow in that product. Temperature control is a very important part of controlling biological food safety hazards. We need to ensure that our food is stored at low enough temperatures where microorganisms cannot grow. It's important to remember that microorganisms are usually not killed by low temperatures, but they cannot grow in low temperatures. The danger zone is that area which is body temperature, where it is nice and warm and bacteria will multiply and go into the log phase where there's exponential growth. This is where toxins can be produced and high numbers of bacteria can grow and multiply. When we cook food, we destroy a lot of microorganisms. Above 75 degrees Celsius, a lot of these bacteria are actually destroyed. To control the pH of food is another way of preventing microbial growth. That's why preservation with an acid or a low pH is so useful in controlling microbial growth. Most microorganisms cannot grow in products that have a very low pH, such as salad dressings, preserves, etc. We can also exclude oxygen. Now, a lot of our pathogens, not all of them, but a lot of them, cannot grow where there's no oxygen. So removing oxygen is actually a good way of preventing microbial growth. We can do this by vacuum packing or canning or even pillow packing with nitrogen gas. Remember that there are microorganisms that can grow without oxygen, but we eliminate a very large group of microorganisms that need oxygen to grow by vacuum packing or excluding oxygen from our food products. We can also apply a combination of these controls. This is called hurdle technology, and a good example of hurdle technology is biltong, which is spiced dried meat that is vacuum packed. Let's have a look at what we've done with biltong. So firstly, we reduced the amount of moisture in the product and therefore reduce the water activity. So a lot of our pathogens will not be able to grow there. We've also added a lot of salt and spices, which further prevents microbial growth. And by vacuum packing it, we also excluded oxygen. So we've applied more than one control measure in order to preserve this product that otherwise would have spoiled very quickly. And that brings us to the end of our course on basic food microbiology. I hope you have a better understanding of food microbiology and that you will be able to apply these control measures to prevent microbial growth and cross-contamination in the food industry.